Hello, everyone, and welcome to today's lecture. Today we are going to be talking about enzyme reactions and some of the kinetics that are involved in this. So this is actually week 15 of our 15-week question-based learning course. So if you want to check out any previous videos, they're all available on our YouTube channel, which I have linked on this slide here. We also have our GroupMe on here, which is where we will send lecture links, as well as where you can ask any questions that you have about any of our videos that you have watched. So definitely encourage you to join the GroupMe whenever we're going to be starting a new course. We'll be posting all that information in there. So if you want to be a part of that, feel free to join. I have our Instagram linked on here as well if you want to check that out. And the schedule of this course has been 2 lectures a week. So one released at 8:30 p.m. on Sundays and one on Mondays. This week was changed due to the holidays, so we pushed it back a little bit. But yeah, so let's get it started. So question 1, we're going to be talking pretty generally about enzymes. So enzymes play a blank role and blank have an impact on delta G for a given reaction. So here we have a classic 2x2 question. So if you know one of these, I would definitely encourage you to use that to your advantage and cross out the other two. Then at least you have a 50% chance of guessing right rather than 25%. So take a minute here, pause the video, and try to answer this question. All right, assuming that you have had a chance to answer the question, I will reveal the answer, which is A, kinetic and do not. So since enzymes play solely a kinetic role, they do not impact the delta G for a given reaction. So we're going to talk a little bit more about what that means and the role that they do play. So the function of an enzyme is basically to lower the activation energy to allow the reaction to occur more easily. So this is not a thermodynamic role, this is a kinetic role. So they alter the reaction rate and activation energy, but they do not affect the total amount of energy that is released. As you can see on the graph here, this thick dotted line, this shows the overall energy release. And as you can see, the blue line with an enzyme and the red without, they both have the same overall energy release. Just the blue line will have a lower activation energy there compared to the reaction without the enzyme. All right, so enzyme structure. There are a few different structures. We're going to talk about all of them, but so enzymes very basically are proteins that have a variety of structures. We've discovered thousands of enzymes, so there's no super easy way to classify them. They all have different structures. They are fit for different substrates. We have a few different hypotheses as to how those substrates fit in. But the classes of enzymes can be broken down into hydrolase, isomerase, ligase, lyase, kinase, oxidoreductase, polymerase, phosphatase, phosphorylase, and protease. So as you can see, the common ending here is ase. That's pretty indicative that what you're dealing with is an enzyme if it has that ending. So there are 2 models that are commonly talked about and that you should know for the MCAT when looking at enzyme structure. So the active site model states that the substrate and the enzyme are a perfect fit. It's also known as the lock and key hypothesis. So neither one undergoes any sort of conformational change after they associate with each other. So as you can see in this picture, it's just a perfect fit and it goes right in and that's it, not much to it. The induced fit model, on the other hand, states that the substrate and active site are slightly different in structure. So when the substrate does bind, it induces a conformational change. in the enzyme that allows it to fit a little more tightly. So as you can see here, these arrows are kind of showing that when the substrate is binding, that is causing those sides to pinch in and make it fit just a little better. So yeah, those are two different models. Yeah. All right, question 2. Now we're going to talk a little bit more in depth about those different classes of enzymes. ATPase is a biologically relevant enzyme present in eukaryotic cells. Which class of enzymes best describes ATPase? So take a minute here, pause the video, and answer this question. All right, assuming you have had a chance to answer, I will reveal our answer here, which is C, hydrolase. So the nice thing about these classes of enzymes is that a lot of them are pretty self-explanatory as to what their function is by their name. So hydrolase, you might have guessed, hydrolyzes. So when you think about ATP and using ATP, it's usually hydrolysis. So that was just one way you could have deduced your way to that answer without necessarily knowing that ATPase is a hydrolase. But we are going to talk about all of the different classes and what their specific function is and how they operate. All right, so a hydrolase, this was the answer to our question. These hydrolyze chemical bonds, as you might have guessed. So some examples are ATPases and proteases. Isomerase, as you might be able to guess as well, forms an isomer, so it just rearranges bonds, and that's what I have a picture of on the right here. So we have malleate isomerase, and as you can tell, the actual structure is not changing. We're just changing from a cis bond to a trans bond. Ligase, these form chemical bonds. So a good example here is DNA ligase. Lyase, these break chemical bonds, but they do not use hydrolysis or oxidation, because if they use hydrolysis, it would be a hydrolase. And if they use oxidation, it would be an oxidoreductase. So a good example here is pyruvate decarboxylase. So kinases, these are enzymes that will transfer a phosphate group from an energy carrier onto something else. So a good example is phosphofructokinase. So if we think back to our glucose metabolism lecture, you will be more familiar. Also, kinase is in the name, so you could probably deduce that one yourself. Anyways, so oxidoreductase, these include oxidases, reductases, and dehydrogenases. So those can be broken down a little more, but just generally they're oxidoreductases. And these will run oxidation reduction reactions. Polymerases, these are involved in polymerization reactions. So a classic example here is DNA polymerase, which is what we have pictured on the right here. So this polymerase is just adding these bases. And then a phosphatase will remove a phosphate group. Phosphorylase will transfer a phosphate group from an inorganic phosphate, and then protease, these hydrolyze peptide bonds. So protease actually also can be in the class of hydrolase. Yeah. So those are all the classes. Here we have a picture just of a protease. See, it's clipping this peptide bond here. And we end with these two separated peptides. All right, question 3. We are going to talk about enzyme regulation. Which of the following describes an allosteric regulation of an enzyme? So take a minute here, pause the video, and try to answer this question. All right, I will reveal our answer now, which here is going to be C. So allosteric regulation, an example is the non-covalent binding of CAMP somewhere that is not the active site. So allosteric means somewhere other than the active site. So you could have crossed out A and D if you knew that. But allosteric regulation is non-covalent binding something in a different site. So phosphorylation would be a different type of regulation, which we will discuss now. So the whole reason why enzymes are regulated in the first place is to maintain health and proper cell functioning, because if all of the enzymes were active all the time, we would have a ton of reactions going on, a ton of pathways running. And as we have gone over before, a lot of those pathways are opposing. So if you're running two opposing pathways at the same time, that's going to be a huge energy cost for the organism, and it's just not sustainable. So the example I have on here is just thinking if glycolysis and gluconeogenesis were occurring at the same time, you would be using a lot of energy and nothing would be accomplished because we would be making glucose, breaking it down, making glucose while we're breaking it down. So you would get no net glucose breakdown or no net glucose production. So yeah, that's pretty much why these enzymes need to be so tightly regulated. And why they are. And there are various ways that they can be regulated. So one of those ways is covalent modification. So this is when you can have covalently attached groups that regulate protein activity, lifespan, and location. So by modifying which groups are covalently attached to an enzyme, you can modify its functioning. So a good example to be familiar with here would be adding a phosphoryl group from ATP to the hydroxyl group of a serine, threonine, or tyrosine. So that is a way to covalently modify those residues. So it adds to the hydroxyl groups on one of these three amino acids, and that alters the functioning of that enzyme. So I would definitely be familiar with those amino acids and that they are commonly phosphorylated in enzyme regulation. So another way is a proteolytic cleavage. So inactive enzymes can be activated with a protease. So we have another enzyme involved here, but basically that, as you can see in this picture, so zymogen, that's the term for an inactive enzyme that is going to be cleaved by a protease. So this blue part here that is cleaved off is called the masking sequence, and then that will then expose our active site and allow the substrate to bind to this enzyme. So another way that they can be regulated is association with other polypeptides. So sometimes enzymes will have separate regulatory subunits. And these can differ a little bit. So some enzymes will require the regulatory subunit to dissociate for proper functioning, and some will require association with that subunit for functioning. So it's not necessarily one way or the other, it's just in general association or disassociation with that subunit alters the functioning of different enzymes. So this was what our question was about, this allosteric regulation. So as you can see, you have an activator here. So this would be the allosteric site where this green square is, because it's a site on the enzyme that is not the active site. So when this binds at the allosteric site, it will cause a conformational change in the active site. And as you can see, that makes it a better fit for the substrate. So that's how that enzyme will be activated. And this binding is non-covalent and it's usually easily reversible. So it's a great spot for like an on-off switch for this enzyme to be activated. All right, question 4 now. We're going to talk a little bit more about regulation of enzymes. Which of the following is not an example of feedback inhibition present in glycolysis? All right, so take a minute here, pause the video, and answer the question. All right, assuming you've had a chance to answer this one, I will reveal the answer here, which is D, fructose 1,6-bisphosphate downregulating phosphofructokinase. So you kind of need to know the order of all of the steps in glycolysis to answer this question. So fructose 1,6-bisphosphate is actually earlier in the process of glycolysis than phosphofructokinase. So that's an example of positive feedback or feed-forward activation. So fructose 1,6-bisphosphate will actually upregulate phosphofructokinase, whereas G6P, ATP, and ATP again are all later products to their counterpart in these answers. So feedback inhibition is usually when a product later in the pathway will inhibit something earlier, telling it that basically we've made enough, we've gone far enough in this pathway, we can start shutting it down from the beginning. So yeah, this was kind of a tricky one. I always struggled with the steps of glycolysis, but I think it's really good to try to incorporate that wherever it's possible to, force yourself to recall that information. All right, so kind of just went over this pretty briefly, but feedback inhibition can also be known as negative feedback. So this is when a later product in a pathway is inhibiting an earlier enzyme. And this makes sense just because once you have a sufficient amount of final product, you'll want to turn off the pathway so you stop driving it towards making more of that product. So this picture pretty simply describes this. So you have D, our final product, is going back and negatively, or it's trying to inactivate enzyme 1, so we kind of stop the whole pathway. Another important thing to note is that a lot of times the most carefully regulated enzyme is that first one in the pathway, just because if that one's off, you're not going to be needing to deal with the further enzymes in the pathway. So that's usually the one that you'll see the tightest regulation on. So feed forward activation, on the other hand, this involves the activation of an enzyme further down in a pathway by a product or a substrate from earlier. So this is basically to ensure that there is enough enzyme later to complete the pathway and drive that reaction to your final product. So this image does a nice job of showing both feed forward activation and feedback inhibition. So yeah, it's pretty self-explanatory, but just wanted to cover that because sometimes the wording can get a little bit confusing. Question 5, we're going to talk a little bit about enzyme inhibition. So an unknown inhibitor is added to a reaction. You slowly add more and more substrate and notice that your Vmax was not affected by the inhibitor. What kind of inhibitor is most likely present in your reaction? So take a minute here, pause the video, and try to answer this question. All right, assuming you've answered the question, I will reveal our answer here, which is D, a competitive inhibitor. So the main thing to get out of this question is that you were able to overcome the inhibitor by adding more substrate, and that is characteristic only of competitive inhibition. Your Vmax will be unchanged, and we will now go and talk about these other kinds of inhibitors as well, and how they will affect your Vmax and Km. Okay, so just briefly now, I'm going to go over it when I go over how these reactions are graphed, but Km is the concentration of your substrate at half of your Vmax. So Vmax is the basically your velocity of the reaction. So it plateaus, and that's just your maximum velocity. And then Km is the concentration at half of Vmax. So the lower your Km basically means the higher affinity your enzyme is for that substrate. Okay, so competitive inhibition, which was answer to our question. This is where the inhibitor will compete with substrate binding at the active site. So as you can imagine, the competitor will closely resemble the substrate in structure. So it can fit into that active site. Even if it's not a perfect fit, it will have to loosely fit in there in order to be a competitive inhibitor. So this kind of inhibition can be overcome by adding more substrate, because your substrate will almost always have higher affinity for that enzyme than your inhibitor. So Km will increase and Vmax will be unaffected. So the reason Km increases is because since the inhibitor is taking up that active site, you have to add more and more substrate to reach that Vmax. So it'll be a slower climb to that plateau, but you will get there. Non-competitive inhibition, this is where your inhibitor will bind at an allosteric site. So the structure does not have to be anything like your substrate. It can be anything as long as it fits into that allosteric site. It is a non-competitive inhibitor. So inhibition here cannot be overcome by adding more substrate, simply because they're not competing for the same spot. So they're pretty independent of each other. So our affinity for the substrate will not change, but Vmax will decrease. You won't be able to reach that state just because a lot of your enzyme will be inhibited due to that inhibitor binding at the allosteric site and preventing some of your enzymes from binding. So uncompetitive inhibition, these also bind at an allosteric site, but they differ from non-competitive inhibitors in that they are only able to bind the enzyme substrate complex. So the substrate already has to be bound into that active site for the uncompetitive inhibitor to bind at the allosteric site. So you imagine that there's probably some sort of conformational change in that allosteric site or something that allows these inhibitors to bind. But just know that that is the key difference. I definitely struggled with remembering non-competitive versus uncompetitive. They sound the same to me. So I would definitely work on committing this to memory, however you can. So for these inhibitors, your Km will decrease because you'll actually have a higher affinity, since these inhibitors will kind of lock the substrate in the active site. So this is a unique case in that your Km actually decreases, and then your Vmax will decrease as well, which you might expect. So mixed inhibition, this is inhibitors that can bind either the enzyme alone or the enzyme substrate complex. So it's a mix of that uncompetitive and non-competitive inhibition. So Km can either increase or decrease, but Vmax will always decrease. So Km will decrease if the enzyme substrate complex has a greater affinity for the inhibitor. So if it closely resembles an uncompetitive inhibitor, and then Km will increase if the free enzyme has a greater affinity for the inhibitor. Yeah, so mixed inhibition is kind of tricky, but I would definitely do your best to try to understand these. And we're going to look at all of these on graphs now, which should make them a little bit more clear. All right, question 6, we're going to talk about enzyme kinetics. The following graph is showing the presence of which type of inhibitor? So since we just went over inhibitors, try to use that to figure this out. I know this is not the most straightforward graph, but if you're familiar with these at all, then you should be able to figure this one out. So yeah, take a minute, pause the video, and answer this question. All right, assuming you have answered the question, I will show the answer, which is B, uncompetitive. So as you can see, the red line is the inhibitor, blue line is no inhibitor. So the important thing to note here is that this is a Lineweaver-Burke plot, and we're looking at the inverse of the substrate concentration and the velocity. So with the inhibitor, since this is moving further away from the origin, our Vmax is decreasing. And since it's moving further away from the origin here, our Km is also decreasing. So as we just talked about, that was characteristic for uncompetitive. So that is basically how you would figure this one out. But we're going to look at Michaelis-Menten plots and Lineweaver-Burke plots and what inhibitors look like on those different kinds of graphs. So Michaelis-Menten plot is pretty straightforward. It's just your substrate concentration graphed against your reaction rate. So as we talked about earlier, we have our Vmax, which is kind of right above where this will plateau at the maximum reaction rate. So Km is our substrate concentration at half of the Vmax. And Km tells us a lot about affinity. So as I said a little bit earlier, the lower your Km, the higher the affinity for that substrate. So here are some inhibitors on Michaelis-Menten plots. So we will see for our competitive inhibitor, we have the same Vmax, but a different Km. So our Km will increase because if you draw a dotted line at half of Vmax, you'll see you hit the black line before you hit the red line. And that's because our Km is increasing. So we have lower affinity here since our inhibitor is competing for the active site with our substrate. So our blue line, this shows a non-competitive inhibitor. So we'll have the same Km. Basically, yeah, if you just draw a line at this new Vmax and then half of that, you'll see it actually falls at the same point as our original Km. But you can obviously see that Vmax is a lot lower here. Yeah, so these graphs, it's kind of tricky to see uncompetitive and mixed inhibition just because you're kind of, I mean, if there's no lines over here, you're kind of eyeballing where half of Vmax is. So I wouldn't worry too much about that. Just know competitive and non-competitive, and then we'll look at uncompetitive on the Lineweaver-Burke plots. So our Lineweaver-Burke plots, this is what was in our questions. So it's the same thing as our previous graph, but we're graphing the inverse of reaction rate and substrate concentration. So your x-intercept is going to be 1 over your Km, and your y-intercept is 1 over Vmax. So those are the points that you're going to pay really close attention to. And then the slope of your graph is Km over Vmax. So with these graphs, you have to pay attention to the fact that as Vmax increases, you're making that fraction smaller. So it will get closer to the origin. So more commonly, when you're looking at an inhibitor, as Vmax is decreasing, you're moving further away from the origin. So even though it looks like it's going up, your Vmax is actually decreasing. The same goes for Km. So as Km is decreasing, the x-intercept moves further from the origin, whereas if Km increases, it will move closer to the origin. So not the greatest quality of pictures, but it gets the point across. So we have our competitive inhibitor here, which shows that they have the same Vmax, which is what we know about competitive inhibitors, but then our Km here increased, so we move closer to that origin. So when they cross at the y-axis, that is a competitive inhibitor. Uncompetitive inhibition, this was the graph in our question. This is showing that our Km is decreasing, so we're moving further away from the origin, and Vmax is decreasing as well, so we're also moving further away from the origin. Non-competitive inhibition, you will have the same x-intercept because our Km is unaffected for non-competitive inhibitors, and our Vmax will decrease, so we will be moving further away from our origin. So definitely do whatever you can to be familiar with this. This is a pretty high yield topic, I would say, for biochem on the MCAT. So yeah, definitely be familiar with the different types of inhibition, what they look like on these two types of plots, and then how Km and Vmax are impacted. All right, question 7, a little bit more on enzyme kinetics. The concentration of a reactant in a second order reaction increased by a factor of 5. How much is the rate of the reaction altered? So take a minute here, pause the video, and answer this question. All right, assuming you have answered the question, our answer here is C, increased by a factor of 25. So the key to this is knowing that a second order reaction means that that reactant is squared. So if the concentration increases by a factor of 5, you have to square that as well to see how much the rate will be altered. So this shows that pretty well here. So a reactant can be either 0th first or second order. So that is this number up here. Whatever exponent these are to tells you what order those reactants are. And the overall order of the reaction is the sum. So if you have K, A, B to the second, that would be like A to the 1, B to the 2. So that will be a third order reaction overall. So reaction order has no correlation to stoichiometric coefficients. So the MCAT loves to try to trip you up with that. So when you're given a reaction and it says, you know, all those coefficients in front of the numbers, ignore them. You just look at the exponents up here. So the rate determining step in our reaction is always the slowest step, and this is the step that determines your rate law. So you only use the slowest step. So I have a little example reaction here. We have A plus B plus C will yield ABC, but this is 2 steps. So our first step is the slow one, A plus B, and then our second step is AB plus C. So our rate here would be rate equals K times A to the X, B to the Y. So you see C is not even a part of our rate because it is not involved in the rate determining step. Yeah, so that's pretty much it, but just try to be aware of that. Another thing I think that the MCAT likes to trip people up on. Yeah, so just pay close attention there. All right, question 8, cooperativity. Hemoglobin and myoglobin play important roles in oxygen transportation in the human body. Hemoglobin, which is found in red blood cells, exhibits cooperativity. Myoglobin, which is found in muscle cells, does not exhibit cooperativity. Which of the following is most likely true? So take a minute here, pause the video, and answer this question. All right, assuming you have answered the question, here is the answer. It is C. Hemoglobin likely has multiple subunits while myoglobin does not. So we're going to go over what cooperativity means, and hopefully that will explain why this is our correct answer. So positive cooperativity is basically when substrate binding to one subunit increases the affinity of other subunits to the substrate. So here in this picture, we see we have one ligand bound in step 2, and you can see here how they pictured those active sites getting bigger. So this is a great example of positive cooperativity. So it's increasing the affinity for substrate in those active sites, allowing step 3 to occur more readily. So that is why for our previous question, hemoglobin likely has multiple subunits, because that is a big component of cooperativity, is having multiple subunits and having binding of one of them, increasing affinity in the other. So negative cooperativity doesn't really fit with this picture, but I tried to explain it like it does anyways. So substrate binding to one subunit decreases affinity. So just imagine that in step 1, the squares are big, in step 2, they're small. So binding 1 then would decrease the affinity, allowing step 3 to occur more slowly. So that's not as common. Usually talking about cooperativity, we're talking about positive, but just wanted to go over that so you're familiar with it. All right, question 9, we're going to do a little review question. So with these, I always encourage you not to look at your notes if possible. If you're really stuck and you can't figure it out, go ahead and look at them. But for me, at least, the best way to study is trying to recall that information. And since it's all fresh, we just went over all this. It's just a good practice to get into. All right, so you have come across an inhibitor that binds irreversibly to its enzyme. Would the kinetics under those conditions be similar to those of a reversible non-competitive inhibitor? So take a minute here, think back to what you know about inhibitors, non-competitive, all our different types here, and see if you can figure this one out. So yeah, just pause the video, take your time on this one, and resume once you've answered. All right, I will reveal our answer now, which is A. Yes, because irreversible binding will cause a loss of active enzyme available for the reaction. So that is similar to our non-competitive inhibitor. When they bind, you have a loss of active enzyme because they bind to the enzyme alone, whereas our uncompetitive bind to the enzyme substrate complex. So yeah, that would be the most correct. So again, something to notice for the MCAT, your right answer is the most correct answer. So it's not a great, not great knowing that, but yeah, you just need to know basically what non-competitive inhibitors look like and how they take away the amount of active enzyme available. All right, question 10, we have one more review question. So fetal hemoglobin has a higher binding affinity than adult hemoglobin. What will be true regarding the hemoglobin dissociation curves? So even though we didn't really cover dissociation curves, we did talk about KM, and that's what you need really to answer this question. So kind of gave you a little hint there, but anyways, take a minute, pause the video, and try to answer this question. All right, I will reveal our answer now. So our answer here is A. The adult hemoglobin curve will have a higher Km than the fetal hemoglobin curve. That is because, as we talked about, Km is a measure of affinity. So a higher Km means lower affinity, lower Km is higher affinity. So that means, you know, since fetal hemoglobin has a higher binding affinity, it will then have a lower Km. And then C is wrong because the fetal hemoglobin curve will be shifted to the left. If you need any help with dissociation curves, feel free to message me or look through our other videos. I'm sure we go over it at some point. All right. Thank you guys so much for tuning in. Again, as I said at the beginning, this is the last video in this 15-week question-based course. So if this is your first one of the course that you've watched, I would definitely encourage you to watch the other ones. I really like the style of this course just because it's good if you already have a little baseline studying done, because then it gives you a great chance to test on how well you know that information, if you're able to answer the questions. And if you're not, then we go over everything that we need for you to get that answer. So it's just a great way to study. So yeah, good luck to everyone in your MCAT prep. Again, my name is Megan. I'm one of the tutors here at Socratic Med. If you have any questions at all about anything, I focus on psych soc and biochem, so feel free to message me about any topics in the group me, and I would be more than happy to help you with that. So yeah, thanks again for watching, and good luck.